Yes, sure. This is what. So, I'm deeply honored for giving me this opportunity to deliver a lecture in this occasion of ADNET and in the memory of Dr. Laljisingh. So I will just speak two lines about Dr. Laljisingh and then I will start my lecture today. So my interaction with Dr. Laljisingh was when I joined in the PhD in CCMB in 1998, and then he took over as the director of CCMB at that time, 1998. And the two things which I remember, that nothing was impossible in during his tenure. And whichever like equipment, if you tell him any problem, if you tell him, he would immediately bring it in CCMB. So we got a privilege working under his directorship. Although I was working, my supervisor was some Dr. Ramesh, but since he was like a fatherly figure, like he contributed lot in the institutional development, and it also reflected what I worked during my PhD. So now I would like to thank the committee for giving me a chance to speak. So today I will speak about the understanding the social language of a prokaryote. Okay. Although our lab works on the plant microbe interaction as the introduction they told. So one part of our lab also works on the plant DNA fingerprinting, because in our institute CDMB we have a DNA fingerprinting mandate. So in that forensic DNA fingerprinting is there as well as plant DNA fingerprinting is there. So I have divided my talk into three parts. So in the first part, I will introduce you to how bacteria can communicate with each other and what we understand by their language. And second part, I will talk about why they communicate. What is the need of their communication? Okay. So bacteria is a prokaryote. So why they communicate and what is the biological significance of that? And third part of my talk, I will tell you is there any problem in communicating? What are the pros and cons of communication? And the fourth part, how you can understand the basic biology of pathogens, particularly the bacterial pathogens, to devise strategies to interfere disease. So I will give you the example of what we have done in our lab. So coming to the different characteristics of a living organism, like growth, reproduction, heredity, homeostasis, response by communication and response to environmental stress is one of the major part of being alive. Okay, so these are different aspects of a living organism. So adaptation to changing environmental condition is very important for a living organism. So this is one of the way in which organisms respond and change themselves to adapt to the change in the environment. And response by communication is a very evolutionary subject. If you consider, for example, the language English, so people say that the evolution of the language is very similar to the evolution in the human evolution. Like it diversifies, as well as there is an analogy in the evolution that explains change or drift towards natural selection. As you see, as you go from one region to another, the language and the dialects change, and they are constantly being evolving. For example, if you see the grammar, the English, for example, in the literature, the grammar has been evolved, but it again sub-evolved based on the countries where it has been adapted. So there is a very parallel to how languages change over time, like human anthropology has changed. If you see different mode of communication in living organisms, there are very nice way, like we are talking in the form of language, but many animals can't talk. So, for example, firefly. Okay, so the firefly blinks in a particular frequency. It actually attracts the mating for the mating. For example, peacock dance, and the most interesting example is honeybees. So, have you heard about the dancing movement of honeybees? So, the dancing movement of the honeybee is to communicate with their fellow mates that where the sweet nectar is there, the flower, so that the whole group can go to that particular field. So there are different mode of communication: visual, auditory communication, and touch mediated, the sensing based communication. For example, in the honeybee. But if you see the life of the bacteria, like it's a unicellular organism, and it is very small in size, 1 to 4 micrometer in diameter. Okay, so this what you are seeing is an electron microscope. It is such a tiny organism. So the questions started arising in late 90s that do bacteria have a social structure like other animals, and whether they can communicate with each other. So very interesting question. Very small organism to very big organism, we know different mode of communication which are shown. Whether in the tiniest organism, they also have a social structure and social communication. So the clue came very indirectly. Okay, so there were some marine biologists who were working on a squid, which is called as Hawaiian squid. So it is found in the Pacific. So this squid actually produces light, like luminescence, in the ocean, and marine biologists think that the production of the light actually helps the squid to find small shrimps in the dark of the ocean. But later on, they got very surprised. The light which was produced by the squid, it is not its own light. It is produced by millions of bacteria that colonizes the light organ of the squid. So it is kind of a symbiotic relationship. So bacteria get sheltered, and the squid uses the light of the bacteria to search for the food. So they obviously got very interested. Now the microbiologists came in. What is, if the bacteria is producing light, then how the light, what is the nature of that light? So they started growing the bacteria, and they got a very interesting observation. They found that the bacteria only produces light when they are high in numbers. So they don't produce any light when they are low in numbers. That means cell number is less. When the bacteria grows, they produce the light. When they are not growing, they don't produce the light. So that indicated that this light production is somewhere related to the density of the population. So they did another very clever experiment. What they did, they took a talking bacteria, that means the producing the light, and they took only the supernatant of the culture, which doesn't have the bacteria, and they put in a low cell density bacteria, and they found that the non-talking bacteria started to talk or produce light. So they found there are certain chemical communication substance which is produced by high density bacteria, and there is a message, this is secreted by the bacteria to tell their fellow mates that there is something that you are how many in numbers, that there is some chemical form of communication is going on. Then the whole field of social microbiology evolved, and people started finding that actually in nature the bacteria doesn't live in a solitary state. It undergoes transition in lifestyle from a solitary to a biofilm lifestyle. And biofilms are very medically important nowadays. If you see most of the hospital infections and this, these biofilms, you see they are made up of millions of bacteria. They form a super complex or a housing complex like structure. Like they have their own protection, the gate, and they make these large clusters to protect them from the other antibiotics or harmful chemicals. So the whole process of signal mediated talking of the bacteria is called as quorum sensing, which is called as density dependent cell-cell signaling. So this is depicted in a very simplified cartoon. When the cell number is less, as the bacteria grows, they start producing the chemical molecules. When the density reaches high, the concentration of the extracellular signaling molecules secreted also reaches very high, and this gives a threshold for the bacteria to master regulate their gene expression and synchronize the gene expression to perform certain tasks which is required in high number. So basically by this method bacteria can count each other and can recognize each other. Okay. So now in the lab you can't listen to the bacterial language, but you can see the language. So we make lots of biosensors which can detect different bacterial chemical signaling molecules in terms of hooking different synthetic circuits. I am not going into much detail. Basically, if that particular language is there, the reporter will blow. So here you see in a plate where lots of bacteria is there, they are talking, and the bacteria in the other plate you see there are lots of bacteria, but they are not talking. In general, bacteria loves to talk like humans. Now, what is their language molecules? So it is as diverse as human language. So the most commonly studied, because this has been most commonly studied, is the acyl homoserine lactone mediated signaling. You don't have to remember the structure. Just I wanted to give you a glimpse that there is lot of diversity in the language. So as you see the evolution of language, like this acyl homoserine lactone, the lactone is the base and the acyl side chains are added. It is very similar to the analogy to the language system. Like every language has a base. For example, Sanskrit is the base and different dialects are added. Okay, like Hindi, Bengali, or like that. So in that way, we vary. The bacteria vary their dialects by keeping the base. So in the later part of the talk, I will focus on this family of signaling molecules, which are important in plant pathogenic bacteria, which we work on, the rice pathogen and the cabbage pathogen. In parallel, there are several different types of quorum sensing signaling molecules. And this is a so sometimes the bacteria are very clever, so they don't speak in one language. This pointer is. So sometimes, like humans, they are sometimes multilingual. Example is Vibrio cholera, which causes cholera. They speak in two different languages. One language is acyl homoserine lactone to communicate among themselves, and one is which is called a borofuranol, which is for communicating with other bacteria which colonizes the gut. So one is their private language and one is their common language. And sometimes they are very clever, they don't speak any language, but they have receptors which are there on their surface that can listen to the language of the others. Very similar to a cartoon where we call it as noisy neighborhood cartoon. When some new neighbors come in, so you are more curious to know who they are rather than them, because you want to know that whether they will adjust in the community or not. Very similar to that, they also try to see the environment, that what are my neighbors and how I have to behave in that. This is a very simplistic cartoon way how the signal transduction process goes on in sensing the chemical signal. So there are synthase which makes the cell-cell communication molecule, quorum sensing signaling molecule, in a density dependent fashion, and there are some intracellular DNA binding response regulator which is called a transcription factor. So they have an affinity towards the signal at a particular threshold. When the density becomes high, the concentration also becomes high, then it binds to the response regulator, and in that way it regulates gene expression. But this is a very simplistic model. It is much more complex. I will tell you an example from our study. So now the main question is why bacteria is spending so much of energy in making the signal. What is its use? So now imagine a situation. There is a bacteria. Imagine it as a person. He is trying to fire bullets to break the wall. Okay, because money or nutrient is there. Okay, so it wants to break the wall. So the firing power of the bacteria is so less that the wall will not break. But if there are many bacteria and they fire together, then there is a great chance the wall will break. So therefore, this is what is the whole concept of quorum sensing. There are certain tasks which is done in unison that will give more benefit to the population rather than if the task is done alone. So you may do the task when you are alone, but the benefit will be less. So this is the whole concept of synchronization of signal and doing the job when you are high in number rather than low in number to get maximum benefit from the source. So in our lab we work on the plant pathogenic bacteria which is called as Xanthomonas, which causes more than 300 different plant diseases. So particularly the rice pathogens, which is Oryza, Oryzicola, which is a very problematic disease of rice, and there are many Xanthomonas pathogens which also causes lots of economically important diseases in various crops. For example, many vegetables, even like fruits like orange, mango. So what we discovered in the lab working with that is that this group of bacteria produces a signaling molecule which is called a diffusible signal factor. So it is a fatty acid like signaling molecule which is cis-enoic fatty acid, and it is produced by the bacteria in a density dependent fashion. Okay, fine. So this is a quorum sensing signaling molecule we identified. But the most interesting part, what happened when you knock out the quorum sensing system in this bacteria, the bacteria completely loses its ability to cause disease in plants. And when you put back again the signaling molecule, it is able to cause disease. So we started looking why a communication system will be linked to the ability of the pathogen to cause disease. So what we started looking, I am not going into much detail. We use lots of genetics, both forward and reverse genetics, and trying to identify the signaling components and the behavior. What we found out that Xanthomonas quorum sensing signaling molecule actually controls the transition from a solitary to the biofilm lifestyle. So this is the vessel of the plant where it colonizes. At low cell density, when the density is less, it triggers the bacteria to do chemotaxis. Okay, so to find its gate or the door, and once it enters inside the door, it triggers the bacteria to produce lots of this cell wall hydrolyzing enzyme, and this cell wall hydrolyzing enzyme will hydrolyze the plant cell wall and release nutrients, and more bacteria will be formed. And when that density reaches a threshold, it tells the bacteria now you form a biofilm by orchestral synthesis of lot of adhesives which are there, which helps the bacteria to attach and form a biofilm in an orchestral fashion. Imagine now a situation, if the quorum sensing, if these adhesives are made at this stage, what will happen? The infection will stop, they will adhere there only and they will not migrate. So therefore this language system has been evolved to orchestra this synthesis of these adhesives and attachment only at a particular stage when the density is more. Okay, so this is one of the easiest ones. So now what are the different so we understand about lot about the biology of the bacteria. I have not gone much into the data because I thought of making a general talk. So these all model how the pathogen to the transition from a solitary to the biofilm lifestyle and how it regulates gene expression and signal transduction is very complex. So, but what we understand from the basic biology that this communication signal is very important for this transition. Can we devise strategies to interfere with signaling system of the bacteria? So there are two strategies. Either you break the signal, they will be not able to talk. Okay. And another approach which we took is the loudspeaker approach. So now in this, for example, in this auditorium, we are doing the lecture. Now somebody plays a DJ music. You are not able to communicate. So here exactly what the idea is. If particular concentration of the signal is important for the bacteria to communicate and to switch on its circuit, we produce more signal. So that bacteria will think we are too high in number, we should stop. This is the loudspeaker effect. So we did both approaches, transgenic as well as non-transgenic approach. So in the transgenic approach, what we did, we took the whole bacterial quorum sensing circuit, engineered it. I am not going much into detail, it involved lot of work, codon bias, corrections, then all the codon optimized, and put it in plants, in the chloroplast, because bacteria is very similar to the in the evolutionary link chloroplast and mitochondria. So that thing was not working in the nucleus when we targeted in the nucleus. So we targeted in the chloroplast and generated transgenic plants. So now all these transgenic plants started producing bacterial signaling molecule in high amount. Okay. So now what happened to the infection? So this is the wild type plant and this is the y-axis is the disease index, the severity of disease. Higher is the index, higher is the disease. So this is the wild type plant showing very high disease. All the transgenic lines show very reduced disease. So what has happened in this case is transgenic plant, when the bacteria attack, we inoculate it. So at the point of inoculation, bacteria has sensed the signal which is produced by this transgenic plant, and that told them now you attach and don't move. You got this point? So basically it is fooling the bacteria that you are too high in numbers when they are not. So this is one of the approach. The second approach, because in India there is a lot of social problem of using genetically modified crops, particularly food. So we thought of developing a non-genetic method. In that we again designed synthetic circuits to produce large amount of these xanthomer signaling molecules in high amount in the laboratory and can be also scaled up in fermentation. And that you can do a formulation with a surfactant and spray in the plants. Just like it is not pesticide, you are basically spraying the signal in the plant and priming the plant, basically priming the plant before the infection. And you see these are the examples. Like this is a buffer inoculated plant and then infected with the bacteria. It is showing disease. But if you pre-prime the plant with the synthetically produced DSS, it reduces the disease severity very high. So this approach now, we are in talks with the Directorate of Rice Research to make it a formulation to use so that farmers can use it. It is not pesticides. It's not. It's just a fatty acid signaling molecule. So these. So what I wanted to tell you that from an example of the basic biology, if you understand the basic biology of the organism, definitely some or other strategies we can think of in solving the particular problem. So I would like to end my talk. I would like to. These are the current lab members. So we are working on Xanthomonas biology. Quorum sensing is one of the major part, and we also work on the iron homeostasis and iron metabolism and mechanism of chemotaxis, how the signal is integrated. We do lots of this phenotypic heterogeneity work also, and this is our institute, the new building in Hyderabad, and these are all my past members of the lab, and I am very grateful for funding from DBT and ACRB and CSI, and also we get core funding support from CCMB. Thank you for your time. Is there any question?
I remember one old talk by answer for pyrophosphates, very important role in
See, I didn't explain in my work. So downstream from the quorum sensing, there is cyclic di-GMP, what we work on. Cyclic di-GMP and PPGPP. So that is downstream. So PPGPP is the stress pheromone of bacteria, like it is PPP, phosphate, PP. So these are made in large amount under different stress conditions, and that is also regulated by the quorum. So in my model, I showed they are there.
all species of bacteria.
No. I showed you, sir, in the beginning of the slide that different bacteria communicate with different languages, different chemicals. So the strategy which we devise will only work for Xanthomonas group of plants and Pseudomonas.
But syntax will be same.
Yeah, the syntax means the overall pathway. Now pathway is very different. They have different receptors, different regulators.
Good morning, sir. This is Dr. Sumit Rawat. Basically, I am a medico working with beacteria. So you just mentioned about Pseudomonas as well. And looking about the DSP approach, one thing we need to keep in mind, like it is not harmful for the bacteria, which is threat. But we are here we don't call it a pesticide. But we have to look at the safety profile once this chemical or this signaling molecule reaches humans or animals. If they will consume the rice, and there will be certain concentration. It will reach humans or animals. So whether it will cause any effect on them or their health, because ultimate goal is to prevent their health. Yeah. So maybe we could study by mice or some other model.
Okay, very nice question. So I will answer your question. Three different. So DRDO, this Directorate of Rice Research in the experimental station. So they have all these ecological and other aspects studied, and whether that chemical will be harmful for the other plant itself. So that part. And the second part of the humans, etc., we have not yet put it in the human, designed it for human. For the agricultural point of view, transgenic we have not released. We have done the demonstration. Only the chemical we have shown that it can work, and that they are doing all the biosafety and other aspects. And for the human use in US and in United Kingdom, the quorum sensing interference strategy is used in hospitals. Okay. So this kind of signaling now they have started to use in cystic fibrosis patient. So cystic fibrosis patient is an opportunistic playground for Pseudomonas aeruginosa, which causes the, the blocking of the lungs and severe hospital infection. And they are multi-drug problems. So these quorum sensing interference strategies are used in John Hopkins and many other hospitals. Thanks.
Insightful talk and in detail talk on how bacteria communicate through language molecules, and if we speak, then why not bacteria? Now, I would like, we are very grateful to have amongst us Shri Laljisingh, who is nephew of Professor Laljisingh, to be here to actually give the award to Dr. Shubajeet Chatterjee. So can I please now request Shri Laljisingh and also the chairs, Professor Satish Kumar, President and Professor D.D. Dubey and Professor Devi Sarkar to kindly come on the stage. I would kindly request Olivia to help with the award ceremony. Shubajeet Chatterjee, can I please have you on stage? Thank you very much. Dr. Shubajeet is also recipient of Shanti Swarup Bhatnagar Prize. And thank you, Shri Arun Kumar Singh, to please come here on behalf of the family to come here for the award ceremony. Thank you for the esteemed, distinguished chairs of the session. And now I would like to sign off and give the next session host, our research scholar, Urgan, to take it over.